Hello, in this video I will be discussing the biochemical basis of the voltage-gated potassium ion channel. Specifically, I will talk about its function within the neuron, a cell in the nervous system. So here we zoomed in into the neuron. We are looking at the phospholipid bilayer of the neuron. So we said that this channel is a voltage-gated potassium ion channel. This means that it's opens and closes based on voltage. So when the voltage inside the cell, in the cytosolic side, is negative, meaning we have more negative ions and negative molecules, and we have more inside the cell, and then we have more positive ions on the outside. There's an alpha helix component on the channel, which at rest, when the channel is closed, will face downwards, and because it's positively charged, it'll face downwards in order to interact with these negatively charged ions and molecules. And so with this conformation of it facing downwards, it essentially causes a conformational change closing the channel. So during depolarization, there's an influx of sodium ions that enter the cytosol. These make the cytosol overall positively charged. So this causes repulsion between the positively charged ions inside the cytosol and the alpha helix, which is also positively charged. As a result, the alpha helix will shift upwards and will face extracellular side of the neuron, and will interact with negatively charged ions and molecules outside the cell via non-covalent interactions. This causes a conformational change that essentially opens the channel. So essentially the driving force of opening the channel is the positively charged sodium ions inside the cell that repel the alpha helix and shift it upwards in order to form the favorable non-covalent interactions with the negatively charged ions and molecules outside of the cell. So potassium within the cytosol of the cell forms ion dipole interactions with water, non-covalent interactions. So upon entering the protein channel, potassium will enter with a hydration shell with water. And in order to pass through the channel and eventually enter the extracellular space, potassium must be able to pass through this filtering region right here. The filtering region is composed of 4 filtering sites, and they have residues which contain carbonyls, which also form non-covalent interactions with potassium. So essentially, potassium, in order to pass through the channel, must be able to break the non-covalent interactions with water and reform non-covalent interactions with the residues which contain carbonyls. So as stated previously, the interactions that potassium forms with water are actually pretty optimal. So how is it that potassium is able to break these interactions, which are optimal, and reform other interactions with carbonyl? After all, they're pretty much the same interactions. It's potassium forming an ion dipole interaction with an oxygen. So what explains this is actually resolvation energy. So the desolvation energy, which is the energy that's required in order to break the interactions between potassium and water, is actually 230 kilojoules per mole, which is energetically costly. So how is it that we're able to do this and it's still an energetically favorable process? The reason behind this is that the interactions between potassium and the carbonyl are actually more optimal than the interactions that are between potassium and water. So the interactions between potassium and water comparatively have a longer distance compared to the interactions we see here with the carbonyl. If we were to zoom in and look at how they're interacting, the oxygen atoms and the potassium ion, we'll see that there's a longer distance in terms of the interactions. So this distance right here between the potassium and the oxygen and the water is much longer than the distance of the interaction of potassium with the oxygens and the carbonyl in the filtering region of the channel. So if we were to invest energy and break the interactions between potassium and water and reform interactions with the carbonyls in the filtering region, we'd actually get our energy back. So although it's energetically costly to break these interactions, we get this energy back when we reform the interactions with the carbonyl. So as we discussed, potassium channel has a region for specificity. It has the 4 sites which are responsible for only allowing potassium to exit through the channel. So within the cytosol of the neuron, we also have sodium, which is another ion. Sodium forms a hydration shell in the cytosol of the neuron. So how come sodium is not able to exit through the potassium channel? In order to figure this out, we'll have to look at energy once again, like we did with potassium. So the desolvation energy for sodium is 301 kilojoules per mole. That's a very high energy cost. For potassium, it was also 230 kilojoules per mole, which is pretty high as well. So why is it that this process for sodium is not favorable? It simply goes back to resolvation energy. The resolvation energy for sodium is much smaller than the resolvation energy we see in potassium. And that is simply because sodium forms much more optimal interactions, non-covalent interactions with water than it does with the carbonyl backbone. So why is that? Simply because of entropy. There is high entropy when sodium is interacting with the waters. This means that the waters in the hydration shell are able to move and adjust, and so they make the distance of the interaction of the non-covalent interactions relatively shorter, which is more optimal. And this also makes it harder to break these interactions, hence the high energy cost of dehydrating sodium. If we were to look at the carbonyl, which has low entropy, the carbonyls within the residues in the selective region of the potassium ion channel are kind of stuck in place. There is no way they're able to move and adjust in order to form better interactions with sodium. Sodium is pretty small compared to potassium. So if we were to look at potassium, for example, the distance between potassium and the oxygens and the carbonyls will be shorter than the distance between sodium and the carbonyls. This means that in order to break the interactions with the carbonyl and potassium, it'll be much higher than these. But if we were to look at the interactions with water, sodium interacts with water and the interactions are much more optimal than the interactions that potassium forms with water. So that's why there's a high desolvation energy for sodium compared to potassium. It's a smaller ion, we have higher entropy, we can form more optimal interactions with water than we can with the carbonyl. So simply, it just would not be an energetically favorable process to break the interactions between sodium and water and form non-covalent interactions with the carbonyl backbone in the selective region of the potassium channel. And Because of this, only potassium is able to pass through the channel. Sodium is not able to pass through the potassium ion channel. The function of the potassium ion channel. It's a pretty complex thing. This channel is extremely selective, and we just discussed how selective it is. But another thing we should know is that the potassium ion channel is a very rapid transporter. It can transport 1 million potassium ions per second, which is an insane number. So it's sort of a paradox that the potassium ion channel can be so selective, but at the same time be a very rapid transporter. So how is that possible? This is possible because of repulsion between the potassium ion channels as they enter the selective region and when they are in the selective region. So this potassium ion we have right here was just dehydrated and is on its way to form the non-covalent interactions with the carbonyl. So as we discussed earlier, the interactions between the potassium ion and the carbonyls and the selective sites are very optimal. Breaking them is energetically costly. So as I discussed, there's 4 selective regions. So this would be one right here, another one, and another one. So in the potassium ion must pass through every single selective site in order to eventually leave the cell. So how is this possible that we're breaking these optimal interactions, reforming them, breaking them, reforming them, and then breaking them again? This seems like a non-favorable process. So what is the driving force for this? The driving force is simply repulsion between potassium ions. They're all positively charged. And since the structure of this channel allows that in the selective region, only one potassium ion can be present at once. So it's a very narrow region. So within the 4 selective sites, only 4 potassium ions can be there. And at any given moment, we would likely not see 4 potassium ions at the same time simply because of repulsion. They would be too close to each other, too much repulsion would be occurring. And they start pushing and shoving each other essentially. So back to this potassium ion right here. It was just dehydrated, and it is on its path to enter the selective region. But the problem is there's a potassium ion right here. And in order to bind to the first selective site, this potassium ion must move upwards. So repulsion forces occur between these two, and what essentially happens is it pushes the potassium ion in the first selective region upwards to the second selective region, and then once again upwards to the third selective region, and then they just keep pushing each other. It's kind of like a cascade, kind of like dominoes falling. Once one pushes one, the other one pushes the other one in front of it, and they just keep pushing each other out. So although these interactions are very optimal, the driving force for breaking these non-covalent ion dipole interactions is repulsion between the potassium ion channels. And because of this, we now understand the paradox. Okay, so let's look at the big picture here. Why is this channel so important? This channel is found in neurons. It is responsible for repolarizing neurons, and it is found in vast amounts in the cell bodies and axons of neurons. Without this channel, repolarization would not occur in the action potential of neurons. In the cardiac system, the channel is one of the main voltage-gated ion channels that regulate conduction, and if we do not have this channel, we will not have conduction within the cells of the heart.